Thank you. Rafael, you quoted two authors, Pace and Denton, who both give an opinion on the uniqueness of the molecular basis in the universe. Of course, the question becomes more and more acute with exoplanets and habitable zone and so on. And we, as astronomers, need more and more guidance to know what we have to look for. How is their position substantiated? Is it an opinion, or, for example, do they give this view because they think, just like with protons and electrons, laws of nature allow only to make about 100 elements, period, and they will make them the same everywhere, in every galaxy, because the laws are the same? Do they have the same attitude that with those elements you can only do DNA and the life property, or do they have another argument, or is it just an opinion?
Of course, it's only speculation based on the only type of life we know. But if you analyze, as I said, it may seem an anthropocentric viewpoint, but when you analyze the atoms that are the same everywhere, you see that carbon, for example, cannot be beaten by any other atom for scaffolding. If you want to construct large molecules, carbon is by far the best. And there is plenty of carbon, and there is plenty of organic matter in the universe. So why would life somewhere different than Earth choose another element? With respect to a solvent, it is conceivable that water could be replaced by another solvent. Why not? And I guess it would depend on the conditions of the particular place where life is developing. Now, this may take us to the definition of life, because what we will recognize as life once we find something, and I think Jose Funes said something in the same vein before. If we want to have a discussion, it will take us forever here to speculate about what life is from the scientific point of view. But for the life as we know it, that requires a liquid solvent and requires molecules and so on, our biochemistry is the best. But I am willing to, I am very open, okay? I'm willing to accept other biochemistries as long as the conditions in other places favor this other biochemistry. But carbon is difficult, I think, to replace.
Does wooden pressure or things like that affect the structure of the cell or create a different type of cell? Like if you live in a place where there is enormous pressure or enormous gravity or something like that, isn't that possible? No, although the atoms are the same, that the cellular structure could be different?
Well, you know that there is life in the bottom of the ocean, 10,000 kilometers, 10 kilometers. Mm-hmm. Where the pressures are unbelievable, and the molecules are more stable because they are bound to solids, to pyrite and other solid material, which stabilizes them. So at least on Earth, life is everywhere, inside the stones and in volcanoes and places where there is high pressure, places where there is low pressure, places where there is high temperature, low temperature. At least our type of life adapts to all types of environments. So again, we have to be very open to accept that life has many chances to develop in other places.
It is a philosophical question. You go against reductionism, and you say you have to see the life as a whole. What is the level of this whole? It is the cell.
I would say the cell.
The genome.
The cell.
Genes, genome, cells.
the cell is the minimal life. And we microbiologists and biologists in general, I think when we speak of life and life elsewhere, we are always thinking, I think, in minimal life. Once you have minimal life, it can develop into more complex systems. But one single cell is already complex. And the big question is, how did the cell start, okay? The first cell start. But for us, there are other biologists in the room, they may answer your question, but in my opinion, when we speak of life, we speak of one cell.
Please, what is the eukaryotic complexity? And is it a measurable quantity? You plot it on the one of your graphs. How is it quantitative, please?
Well, you know, life comes in two types of cell structures. The prokaryotic cell, which is very complex, as I showed before, but the eukaryotic cell is much more complicated, much bigger, at least 2,000 times bigger, and it has supramolecular structures and organelles that the bacteria don't have. It is much more complex. It is difficult to quantitate how much more complex it is, but a eukaryotic cell may contain hundreds of mitochondria, and each mitochondria supposedly comes from a bacteria. So, and there are many bacteria that live inside eukaryotic cells. So it's a world for a bacteria to go inside of a eukaryotic cell. It's order of magnitude more complex, but it will be difficult for me to quantitate in terms of size and structural complexity.
So I want to ask a little bit more about these fascinating experiments with the yeast strains. So what you told us was that you could take out one by one 80% of the genes, but I just want to make sure that means you take out one at a time and the cell still works. You can't take out 80% of the genes and the cell still works. But for 80% of the genes, you can take one of them out and it'll still work. What's the definition of it still working? That is, there must be some functional definition of what it means to say that a gene is essential or not. What was the criterion?
Okay, the criteria is that the cell is not viable if it lacks the gene. Okay. You mutate the gene, and if the cell doesn't grow, the gene is required. But what we have learned in these knockdown experiments is that the genome is very plastic. So you delete one gene, and other gene with a corresponding protein will replace that function.
Okay,
this is amazing. It's amazing.
So surviving was the criterion, not necessarily whether it makes my bread rise.
No, survive.
Okay.
Surviving, yeah.
But should one qualify this statement by saying surviving in the very mild and pleasant conditions, we tend to grow them in the lab, as opposed to outside, submitted to whatever stress and difficulties
you are completely right. And that what you are mentioning now applies for the experiment with the mycoplasma that I showed, because when you want to find the minimal genome, you have to provide the cells with all they need in order to have the minimal genome. You cannot force the cell to make an amino acid or a vitamin or whatever. Yeah, you're right. Good point.